So up until this point, we've been working only with this idea of dominant versus recessive, but there are things that fall outside of this very simple framework. So now we're going to talk about variations on Mendel's law. So Mendel's law are valid for all sexually reproducing species, but a lot of time genotype does not dictate phenotype in this simple way. What if, for example, we started with a red flower and a white flower and we crossed them together? Based on our previous understanding of Mendel's law, we would expect all of the flowers to be red, right? Since red is dominant to white here. However, sometimes what happens is instead we get a hybrid or a mixing of the colors. This is a new phenotype, and it's caused by something called incomplete dominance. Incomplete dominance happens when one allele of the dominant gene is not enough to give the dominant phenotype. In this situation, you will end up with one red, two pink, and one white. When incomplete dominance happens, the phenotypic ratio is going to be the same as the genotypic ratio. In this case, that's 1 to 2 to 1. Another time when this kind of Mendelian genetics breaks down is when multiple alleles are expressed at the same time. This is what happens with blood groups, for example. The ABO blood group is actually made up of 3 alleles, not 2, and they are considered co-dominant, meaning that both the A and B alleles are expressed at the same time in heterozygous individuals. This happens because these alleles determine the shape of carbohydrates that are present on the outside of red blood cells. So if you have the A allele, you will have the A carbohydrate. If you have the B allele, you will have the B carbohydrate. And if you have the A and B allele, you will have both. If you're homozygous for the I allele, you'll have neither. These carbohydrates allow your cells to recognize which red blood cells belong to you, and they also dictate which kinds of blood you can receive for transfusion. So for example, people who have type A blood can only receive type A or type O. They cannot have B or AB blood. Type B blood is the opposite. They can receive type B or O, but they cannot receive A. Type AB can receive anything, and type O can only receive type O. So let's think about this in the context of a problem. Maria has type O blood, and her sister has type AB blood. The girls know that both of their maternal grandparents are type A. What are the genotypes of the girls' parents? Let's start by building a very simple pedigree. In this pedigree, we have the girls, their parents, and their grandparents. The first thing we want to do is we want to put on the board everything that we know currently. For example, we know that Maria is type O, and we know that her sister is type AB. We also know that both of their maternal grandparents are type A, and we want to know what their parents are. Now we can think about which genotypes we already know. So for example, we know that the only way to get type O is to have 2 I alleles, and the only way to get type AB is to have 1 A allele and 1 B allele. So we can go ahead and put those on the board. This means that Maria's parents have to have each given her an I allele, and her sister must have gotten an A from one parent and a B from the other. This can only happen if each parent is heterozygote. One of them is AI and one of them is BI, but which one is which? This is where the grandparents come in. We've been told that the maternal grandparents, i.e. the parents of the mother, have type A blood. This means that it's very likely that their daughter is type A. Therefore, we would say that the mother is AI and the father was BI. Up until this point, we've assumed that everything that we see is based off of a single gene, but that's often not the case. Many characteristics result from polygenic inheritance. This is where a single phenotype results from the additive effects of 2 or more genes, and this is very common. An example of that is human skin color. Human skin color is controlled by 3 different genes, in this example A, B, and C. The number of dominant traits that you have for these 3 genes determines the color of your skin. This means that it is highly variable, and in fact you can see that if you looked around your classroom earlier in the semester. This also means that determining where someone lives based on their skin color is actually pretty difficult. For example, if you were to conduct an F1 cross with these 2 individuals that are the same medium shade, you would get all of these possible colors. So just because 2 people are the same color doesn't mean that their kids will be the same color as them. Alright, so now we're going to tie all of this back to the idea of chromosomal inheritance. So the chromosome theory of inheritance states the following things: that genes occupy specific loci or physical positions on chromosomes, and that chromosomes undergo segregation and independent assortment during meiosis. Mendel's laws correlate with chromosome separation in meiosis, and we've already talked about meiosis previously in this class. The idea here is that there is equal probability that chromosomes can line up in one way or another. So for example, when that happens, depending on how chromosomes line up, that's going to change how different alleles end up being inherited, and that this is the basis for things that we see in 9 to 3 to 3 to 1. So when we're thinking about what gametes are going to be produced by an individual, we have to consider all possible arrangements. There are, however, of course, examples. Sometimes we don't see a 9 to 3 to 3 to 1 ratio, and this happens when genes are linked. Linked genes means that the genes exist on the same chromosome and that they tend to be inherited together. For example, if genes are located on the same chromosome, when meiosis occurs, they will still end up being together. This means that instead of seeing the 9 to 3 to 3 to 1 ratio that you would expect from this dihybrid cross, you will instead see a 3 to 1 ratio, which looks an awful lot like the way a single gene behaves. The exception to this is when crossing over occurs. We talked about crossing over when we discussed meiosis. Crossover events can occur between linked genes, so you can get recombination events, but these are pretty rare. You can calculate this frequency as recombination frequency. I am not going to require you to calculate any of these distances, but I want you to understand what a linked gene is and that it behaves differently than genes that are on opposite chromosomes. We can also use information about recombination rates to tell us where genes are physically located on a map of a chromosome. But for our purposes, we are going to assume that crossing over never happens. If you're interested in recombination frequencies and how crossing over works, I recommend that you take a class on genetics where you will be taught how to calculate this.